It's going to be a very broad discussion on relationship predictions and lots of different aspects about both autosomal DNA, and Rob's going to talk also about Y chromosome predictions and some of that. So it's going to be kind of a fairly diffuse and broad range of questions and topics we have for the panel. We've kind of all contributed to it to a certain extent.
I'm the odd man out here because my wife would agree that I mostly live in the Stone Age, which means I'm more interested in Y chromosome, especially because the data is so incredibly rich for monoparental. My interest in genetic genealogy is mostly monoparental and specifically Y chromosome because there's just so much data. So you might, my tiny bit of notoriety in this, I'm the author of SNP Tracker. That's not the only app. I have a half a dozen web apps out there, Britain and Ireland Mapper and a SNP Tree Explorer, and they're all free. I'm a retired scientist, and you can find them all at that URL on the bottom left, which is a funny name. I just left it there because I'd had that for years for other business purposes. Just a very quick thing about my point of view of this. My own thing as a retired data scientist, chemist and all, is as a hobby, I view my life here is defined by the data source. It's not so much what I want to find out, it's what the data sources that are accessible, legal, available, legit, high quality, what they allow, what fun they allow you to have. To me, it's all about data sources. Here's my little world of stuff. So I don't view any boundaries on data sources other, of course, than access and legality and privacy. But I don't keep a database at all. So I don't have any privacy types of concerns at all. I love the fact that FTDNA makes their Y haplotree public to a great extent, also the mitochondrial haplotree also. I'm happy to nip into archaeology and history literature. I started out doing this 5 years ago when I retired playing with STRs. They're kind of passé now, but I can cluster those very efficiently. I have done and played a lot with surnames because I was able to get a hold for Britain and Ireland. It's not that I'm especially Anglophilic, but that's just where the data is. And of course, I speak the language. So you can do a lot by mashing that up in so many ways with a Y haplotree. You can do actually quite a bit in terms of locating people's historic Y ancestry path. Some other things I haven't put out, they're kind of in beta. I have a GEDCOM Explorer application. I can easily handle 5,000 names in it. I can go up to 50,000 with all kinds of nested graphics, DNA, relationship predictors, simple stuff. I mean, simple centimorgan predictors, just taking the means of normal curves, but any kind of DNA you want. And another thing where I have experimented with tapping into WikiTree, which is a very good API, and I can map all your ancestors on maps from that. But anyway, just to note, since we're talking autosomal DNA today, just thought I'd make a plug here for just the differences in a very simple-minded point of view, probably everybody knows, is the time scope. My metaphor here is that autosomal DNA is a candle. It shines its light in all directions. It only goes a short distance, but that Y DNA or mtDNA are laser pointers, illuminate a very small amount of the universe, but they go an enormous distance. So again, autosomal poops out, roughly speaking, about 1800. People born 1800, but the others easily go back to the Paleolithic and the origins of our species. 240,000 years is no problem. And then finally, maybe I'll dwell on this for just a moment. It's the way I see bringing this all together. To me, we're all doing the same strategies, just at somewhat different scales. Autosomal DNA, I see as a little yellow box in the lower left. That's you or me, and the little red icon there in the lower left. And your ancestry is going straight up vertically from you. And after an ancestor or two, you hit a question mark, and that's the proverbial brick wall, the unknowns in your ancestry. To me, the philosophical level, the metaphoric level of ADNA, autosomal DNA, is it allows you to jump sideways to other people you didn't know before, and this would be your archetypical second, third, fourth cousins. And if you're lucky and they're responsive, you find out they have a paper trail that fills in blanks in your paper trail, and then you can bring that together and discover common recent ancestors. Well, I don't see any philosophical difference between that and using MT or Y DNA. If I go out a little further, which I do in the time reach, in those, I can find a cohort of people, and the dark little people icons mean they probably have real names. The hollow icons of people mean they don't have names that you're not going to know, or they didn't have names, surnames. And then we don't just use paper trail. We rely on things like haplotrees, surnames, locations, paper trails, or for the really distant relationships, we rely on archaeology, history, and ancient DNA. And those allow us to find common ancestry for clan, we'll call clan/family in a general sense, maybe 1,000 years ago. And if we go back before surnames arose, we can easily get back into, typically this takes form of migration history of the great, of Homo sapiens 3,000 more years ago. So that's where I live. And that's a really quick summary of what I do for a hobby. And back to you folks. Thanks.
Thanks very much, Rob. Dave, maybe why don't you go ahead and unmute yourself. You said you had a few comments you wanted to talk about as well here. So why don't you speak up for a bit?
Okay, great, Tim. So I wanted to start off with a little soliloquy on innovation in genealogy, because we're seeing really good examples of that at the GFO SIG that we've been coming to. They really are one of the few groups that really has featured a lot of innovation, really cutting edge stuff. And Brit's a good example of that today. But I wanted to mention a few problems that exist there. One of them is that there are very few places that actually help us incubate ideas and innovations, and we should have some of those in genealogy. There really are some cultural issues that are involved here too, because I think in many ways our culture in genetic genealogy is broken. And what I mean by that is that the business interests often trump collaboration opportunities, that the data is locked up very frequently, and when you can get it, it's not so well structured. The innovators, I think, are underrepresented at conferences, even things like Roots Tech, which I think is something that could be addressed. But even more concerning is that some of us innovators have been stigmatized rather than supported by our colleagues. It kind of gets you back to the Peter Drucker old saying of culture eats strategy. But in genealogy, we really even lack the strategy, and I think there would be real opportunity to improve in this area. And Tim, you've brought this up in previous sessions of this group, and I hope you can maybe help create some of these opportunities for innovators. So back to today's subject of predicting relationships, I wanted to kind of speak about three areas here briefly. I'll talk about the data, about how we might bring in different data types like Rob just mentioned, and a couple of new approaches, including things like graphs. We need better and better organized data. So I think we've learned today that the current methods really have insufficient data to do exactly what we want. And I would commend Brit for trying to get that data and improve the analytics. It is hard to come by. And there's a number of reasons for that. One of them is the informatics standards that we have are not very good. GEDCOM 5 is terrible. I know there's a GEDCOM X, but most people don't use it. Haplotrees, as Rob just mentioned, are available. They're in computable format, but the tree themselves can be a mess. And we have almost no standards for evidence in genealogy or in the legal community for that matter. I'm a physician, and we have pretty good standards that get into things like ontologies and vocabularies and document structure. Those are really important things if you're going to start trying to do robust informatics kind of work. There are examples of some good tools out there, APIs that exist in certain places. But main point here is that what Brit is trying to do today should be easy. You should be able to get this data quickly, easily, and it's not possible today, and it technically is possible. Collaboration is important. There are very few APIs out there. I didn't put in WikiTree here, but Family Finder has one too. The result of not having these standards is tool developers have to scrape data off of websites. I've chosen not to do that because I think it's a very bad practice. And if anybody's been on Family Tree DNA, you notice the tremendous changes in performance of that depending on what time you're on. I think a lot of that, I don't have hard evidence, but I think a lot of that probably is from other people scraping those websites and interfering with the production servers. I run an operation called Graphs for Genealogists, and I took Brit's challenge. Can I produce a report which would give him the kind of data that he needs? And it's a kind of a plea for other developers to think about how you can develop things that help our colleagues. So I'm going to do a series of 3 slides. This is the report that I can produce in a matter of a second or two. The first thing in trying to do matching is you have 2 people. You have this person here with their identifier, their sex, their birth dates, and another person here. And then you have what side of their family are they're matching each other. And there's all kinds of possibilities that exist for that. So with that, you can then take it to the next level. Really, we have what I call a knowledge graph, which means you have many different kind of nodes that are connected together. If you want to read about that, you'll get this slide deck. There's an endogamous blog that really discusses knowledge graphs in more detail. This gives you now the centimorgans, the total autosomal, the shared centimorgans, the longest, the shortest, the number of segments. Does the same thing for the X chromosome. And because we have the family tree in there, we can actually say, is it possible that these people have an X-linked relationship? And as you can see here, there's a couple of examples where they don't. And in family, this is a little bit not representative. My experience with family tree DNA, that this is about 5% of the time they report an X segment, and it's not possible. The other general rule of thumb is that in X chromosome, we should kind of use the threshold of 15 centimorgans or more, which applies here. But if you look up here, we've got two matches that do have a distant relationship, and we have a small segment there. So there's some argument about that 15 centimorgan. So here's actually the meat of what I wanted to get to. If you have a family tree, and I'm using this as an example, this is me and a distant cousin, and we have three common ancestors. This man who married sisters. So he's a common ancestor, and so sisters' parents are a common ancestor. But the way you can deal with this is to count the number of hops in the graph. And in this case, both of us have 5 hops to this one, and we have one common ancestor, which you can represent like this. And you can then look it up. That's a half-fourth cousin. Likewise, these two, you have this representation. So it's very easy to calculate the relationships in a graph by just counting these hops in the graph. And what you see here is that in this case, we've got a number of people who tested in this scenario. This is me, but there's other cousins here who have tested as well. So we have a number of different possibilities here. It's also very easy in a graph to calculate the coefficient of relationship. Now, if you just have one common ancestor or two common ancestors, that's a relatively easy thing to do. It gets a little more difficult if you've got 3 or 4 or 5. 6 common ancestors. And in endogamy things we've got people who have many common ancestors that are shared by 2 people. So the coefficient of relationship is a number, which I think in many ways is going to be better than these descriptors. But even more important, and this is something Brett brought up today, is the pathway to those common ancestors. So I'll use this as an example where I've highlighted this is the M is maternal, the P is paternal. That would be this is a father here. That would go as a paternal relationship. And if you have 3 common ancestors, you're going to have 3 different paths. And in this case, the first person in the match pair has this, and the second one has this. And what you see is they all arrive at the same place with the PMP, but the path is entirely different. Just look at the number of maternal links here compared to the paternal links here. So how is that going to be used in trying to compute, and why is that important? Well, Brett gave us a very good discussion of that. There's a different crossover rate in males and females. So you're going to expect a different number of segments depending on the pattern of the path that's involved. Those probably are going to account for a large amount of the difference that we see, that fifth cousins are not going to look the same if the path is different in terms of their shared centimorgans and the shared segments. And I like to advocate for the coefficient of relationship as a better way to represent relationships than just the nomenclature we're now using. And I think I can answer yes, that I think this graphs for genealogy solution provides a better set of data. But I want to give you one other perspective on this and how to look at this, because instead of trying to predict things, can we actually get to a more concrete way of doing this? Now, I live in the Midwest where we have lots of corn, and I like this analogy of the field of dreams. Wouldn't it be nice if we could have our ancestors walk out of the cornfield and give us a sample of their DNA? And that's what we can do if you create avatars, which are really in silico or in computer reconstructed ancestors. And to my knowledge, I think Graphs for Genealogy and Kevin Borland, who spoke earlier at this group, are the only ones really doing this. And what that provides you with is, they say, your great-grandfather, great-great-grandfather's DNA segments. Those segments you can now match to your the people you have in your data. And that's a very clear way of indicating that that person is in the line of that great-grandfather. In the graphs, you can look at this also a little differently. And what you would expect from theory is that the coefficient of relationship would be linearly related to the shared centimorgan. And if you look at this graph, it looks very nice. The problem is you have a whole lot of stuff clustered up here at the bottom. And if you look at that, you will see, and you plot that on a log scale, so you can pull this apart. You see that you do have, on closer relationships with higher CORs, a linear pattern here, but then things really level off here. And you can begin to see some outliers here. So these are not normally distributed scenarios. Here are these people who have basically the same COR, but they're 1 or 2 orders of magnitude different in terms of their shared centimorgans. The question is, what's going on here? Are there sticky segments? Is this a selection bias, which, having listened to Brit today, that certainly seems very likely? Or is there something else going on? And I think there are explanations for this in Mother Nature from what we know about biology, but also in what we know about graph theory and graph mathematics, which we don't need to dig into today. Turn it back over to you, Tim.
Great. Thanks, Dave. I think we're ready to just go ahead and jump into some of the questions. We have a fair number, and we can spend as much as maybe 5 minutes on any one question if we want to do that. So first question we would have for our panel today is, how much precision can we expect in terms of relationship calculations at various different degrees of genealogical relationship? I know we've kind of touched on this today, but let's maybe get our thoughts from the panelists regarding that issue.
Yeah, I can chime in on that. So you can see at very distant relationships that they could be off by 2 or 3 percentage points. You can see sometimes the order isn't exactly what you would expect. For example, you go down the list from most probable to least probable, you might see that they don't go 7th cousin, 6th cousin once removed, 6th cousin, 5th cousin once removed. There might be 1 or 2 out of order, and that's because those predictions, the probabilities are so low, less than 10% a lot of times. If they were off by 1 or 2 percentage points, the order could get switched, and you can see that happen. But I think for high centimorgan matches, they're probably off by the same amount, maybe 1 or 2 percentage points, which is pretty much nothing for an 80% probability. If you have a paternal grandchild prediction of 90%, and that's the correct relationship, all the other possible relationships add up to 10%. It doesn't matter if it's actually 89% and 11%.
So I have a couple comments here. More data is helpful. We mostly focus today on segments and shared centimorgans. But if you bring in the other data, like Rob mentioned, if you have the monoparental data that has got a Y or a mitochondrial match, you're in that line. If you have an avatar who matches somebody, you know what line you're in. And you've talked a little bit about bringing in other data too, Brit, like the age. I would add geography to that as well. I have examples of relatives who have mitochondrial matches going back to late 1700s. And I have some people then that I don't know, but they're matching, and I know that they lived in the next county over. So that geographic information really makes it much, much more likely than the matches in Norway with that, mitochondrial haplogroup. So being able to combine more kinds of data together is going to, I think, help a lot with relationship prediction.
Absolutely. I couldn't agree more. And I'll bring up another topic that may come up again later, but aesthetic to what Dave was talking about, call us amateurs if you want. It doesn't mean we're not expert. It means we're not engaged in a corporation doing this for profit. And that is that, one of the elephants in the room is Ancestry. They're Ancestry.com. They're also Ancestry DNA. They've published, I think, some brilliant papers in Nature where they've combined hundreds of thousands of bits of aDNA databases as well as their family trees, which are imperfect, but they curate them at massive computer scale. And when you put those all together, they can solve enormous problems. So to me, maybe it's discouraging in a way, I suppose, for the earnest amateur. The future is going to lie with the people who can integrate these massive data sets on a huge scale. And the vendors can do that now. They already have user-entered trees. They already know my problem before. You have unknown A and unknown B. They know the data between A and B at the segment and chromosome level. They're running those calculations. And back to data access, we could debate later about privacy and openness and all that. With great respect to Brit's, a beautiful example of fine-grained precision in thought, but he's limited ultimately by the data. And so if somebody else like Ancestry brings in all this other stuff, geography, all that, there's no way you can do better than have that orthogonal information.
So the other point I could make about this too, there's kind of two approaches to this. One is simulation, which is what Brit is really very good at, and Rob also. And then there's the in silico, where you're actually trying to use the data to create a knowledge environment that's based on actual facts. And one of the advantages of graph databases is you can connect together a lot of these facts. And instead of simulating or estimating, you actually can connect the dots and come up with, I think, more precise answers. You could look at my great-great-grandfather and my matches to that person, and you could guess and simulate and use the kind of analytics we heard about today. But if you actually have a DNA of that ancestor and you can match it to that match, that's a much more powerful kind of methodology. So they both have value. But I think I just wanted to call out the difference between simulation and in silico.
Yeah, and reproducing the amount of DNA that your ancestor would have shared with your matches is really important, and hopefully we're doing a lot more of that in the future. I mentioned earlier, how useful the Watto tool can be with the probabilities of multiple connections, but it's multiplying probabilities times each other and assuming independence, because you have to assume independence to multiply those probabilities times each other. But I've already proven that if you have a sibling tested and you stick them into your Watto tree, you actually hurt your predictions more than you help. It would be better to always leave them out than to enter them, because it's not independence. You and your sibling have related correlations with how much DNA you'll share with the match. If you share a lot with that match, your sibling or half-sibling likely does too. They're more likely to share a lot because you do. Really what we need in the future is tools like Watto. Instead of entering multiple siblings, they need to automatically figure out that in silico amount of shared DNA your ancestor would have had with the match.
Yeah.
Exactly. It's an opportunity.
Okay, thanks. I think this kind of leads into the second question. I've kind of touched on this a little bit earlier with some of my comments, but the next question is, how does having multiple relatives tested help improve our relationship calculations?
So I could comment on that too, because that is my forte. I have projects in various family lines where I may have 60 or 70 kits, all of whom I have their DNA data in there from family tree DNA. What that does is you now have in that database over 250,000 matches with which you can start to do your analytics. And that creates a very complex graph topology. And that topology can be used to do what are called agglomerative clustering, which is a hierarchical clustering. So when you do that, you can easily line up the people with a branch of your family tree. That works very well for close relationships. It's still a bit fuzzy when you get to more distant relationships, but you can get even to that level. I've got clusters that I can definitely relate back to great-great-grandparents using those methods. So having more data is definitely good. The problem is it takes new methods to manage that kind of data. I think personally a graph database and a graph environment is going to be the future of that. I mean, if you look at where industry has gone, I mean, Google and all of the search engines, they're all working with graphs and not with relational kind of systems.
I'm smiling because I'm having fun at Dave's expense in my head here. I do that. Again, 5,000 is no problem for me in parsing GEDCOMs and doing graph traversals. And I'm always in graphs, but I don't use a graph database. I do it in JavaScript, which is a fast language. And you get the same results.
Yeah. Well, actually, the graphs that I work with, you're doing with Java code behind the scenes.
JavaScript.
Yeah.
Right. No,
yeah.
I'm doing it real time in your browser.
Yeah, I know that. Yep.
Yeah. I can all go open source too. Anybody wants my code, you just ask.
Brett, do you have any other comments on that, or we good on that topic?
Dave, I should share a GEDCOM parser with you, Dave. I can make you a little tool. You drop any GEDCOM on, it hasn't failed on me yet, and it'll spit out any structured JSON object you want.
Oh, no, I can do that too. I parse them and put them into nodes and relationships in the graph database. Yeah. Yep.
Yeah, just one other thing related to what we've discussed. I would love to see, in a predictor entering the amount of X DNA you share with a match, X chromosome.
Sure.
And not only is it taking into account the difference in, how much DNA you share on an X chromosome match, but it's also eliminating all the relationships that aren't possible. I think that would be very useful. As David showed, though, sometimes, I think you said 5%, people are matching on the X chromosome when they shouldn't. Actually, in that survey I created, first I asked how much X DNA is shared, if any. And the next question is it possible to share X DNA with this match, which is kind of contradictory, but what I'm after is, are you sharing X DNA through some other line or side of your tree than you should be based on your known connection? And I wonder if that can help us with statistics on how often for our more distant matches with more than one segment, maybe that can help us figure out how often a match that we think is related in one way is actually related in more than one way.
Yeah, that's possible to do that, Brett, in the graph database. It's very easy. What you do in a graph database is as you traverse the graph, instead of collecting you collect the gender of the people and get that concatenated MP string, and then you just say, I want to get rid of anybody who has a PP where there's two men, and immediately you just get the X inheritance pattern. And so that's how I can tell you whether somebody has a possibility. It's very easy to do. And we've got the centimorgans for those people as well. So what I haven't done yet, which would be easy to do, is to do a correlation of relationship based on the X, because there it's not just 50%. It's 50% if it's a mother going, or a woman getting their stuff, but it's not when for a man to a son is 0. So, and for a man to his daughter, it's 100%. So, it's a little different calculation that way, but that would also be important in what you're trying to do, Brett, in terms of the probabilities.
Right.
Because a father to daughter is going to be a much more predictive thing than a mother to a daughter.
Okay, Tim.
Well, that's kind of tell already. I think we've covered one of the questions about the role of the X chromosome. Let's maybe lead off with you, Rob, on this next question.
Yeah.
How can YSTR and SNP testing results improve our relationship calculations? Right. And we can maybe throw the mtDNA in there as well, because they go up both different lines of our tree. But maybe go ahead and give us your perspective.
Right. Well, I'll give maybe the perhaps the non-obvious answer first, and it applies to all of them. Is its exclusion is incredibly powerful, and that is most of these tests, because basically mtDNA. I'm being very general here. mtDNA and YSTR DNA are so limited in their amount of data they carry for a variety of reasons. Basically, the sizes of the DNA bits involved is that they're kind of poor for inclusion, especially for STRs under the large R1b haplogroups. The statistical variance of STRs overlaps what you'd expect from inheritance. So you can't tell anything. It's called convergence artifacts, a terrible term, but it's a mess. But what it can do is exclude. If you want to think about it, this is the use of forensic DNA. Forensic DNA doesn't tell you who's guilty. It tells you who's not guilty with tremendous power. And so you can have even a Y12 STR, which is the old standard, inexpensive old standard for STR. And it may not tell you who's you're related to, but it tells you who you're not, because it's very powerful in that negative sense. So maybe others could chime in. I don't think we pay enough attention to the power of strong negative information in our tools or even in our thinking. But it saves a hell of a lot of time if you can, eliminate a whole set of branches of your building family tree that you don't have to pay attention to now as you work through more subtle calculations. So there's a general answer. We can come back to why SNPs, which is a completely opposite almost. It's the gold standard for definitive paternal relationships. But we'll leave that for a little later, maybe.
Well, I think the other, can get even more complicated, because I have an example of my Stinnett family, which comes from colonial Maryland in the 1600s. And we have autosomal matches for many cousins coming down from that branch. But we have two Y haplogroups. One is in the I, one is I, and one is R. So there's somewhere back in that colonial time, there's a woman who mated with two different people. One is in the Calvert line, which is the famous Lord Baltimore of Maryland, and the other one is mine, which is less prominent. But yet they have, going back that many generations with the split occurring back then, we still have autosomal matches with the people who have tested.
It could be something different, of course, too. It could be in a prehistoric, and you got two named descendants coming over who are unrelated Ys. But I don't know.
Then why do they have the autosomal match?
So you're probably right. It's probably colonial NPE.
Yeah, but this brings up the other case here. I mean, we have cousins who are tested here, but we don't have the 23,000 descendants who have tested. So I still think there's sticky segments here. It's not just a matter of selection bias, because it's true that we're likely to have one match that far back. The odds of one of those 23,000 actually doing a test is small. So that's where I guess I'm still thinking that we do have sticky segments, and there is a biology there that is now emerging that I think may explain this. We could go into if people want to know, but there are things that are conserved in genetics that don't get enough attention in genealogy.
I think there's a lot of interest in that whole topic. I think one of the things I've observed is what happens is that there are sometimes very prolific ancestors who left a lot of descendants. You could look at people like Richard Warren, for instance, who came over in the Mayflower and is estimated to have at least 2 or 3 million descendants. And if you happen to descend from him and you happen to have inherited one of the segments that he brought to colonial America, then you may be matching thousands of people on that particular segment, given how many descendants he had. And so I think Brit's comments in his presentation about population weights and things like that really comes into that, because particularly when you're dealing with colonial America, many couples had 10 children or more, and oftentimes those children had 10 or more children, and in various different populations, like the Mennonites, like in my case, we have very, very large numbers of descendants of some of these people who lived only 200 years or so ago. So there's a lot of confusion over this whole, quote, sticky segment thing. I prefer not to use that term because I think it kind of confuses the whole centimorgan thing. I would rather like to stick with centimorgans and just realize that sometimes these segments have higher frequencies, either just through luck or through a very prolific ancestor who left a lot of descendants.
Well, yeah, I coined the term monophyletic segment because it gets around the issue of sticky and all that. But what I mean by that is it's coming from a single ancestor. To me, that's a better term. I'll just mention a couple things about the biology here, because we have focused on segments that are mutating rapidly, and because that gives us the kind of data we need for these close relationships. But there are parts of the DNA that are very well preserved. We know that for certain regions, like the HLA groups and things like that. But they're now, in the last couple of years, there's been a whole new thing described in DNA called topologically associating domains, or TADs. And these are little 800 base pair segments that two of them will match up on the DNA and start to form a topology to the DNA. So our chromosomes are actually not linear, they're actually spherical. The importance of these TADs is that they are very highly conserved. They do not mutate very frequently. So I think we're going to start seeing the introduction of some new segments that we're going to be able to get that will allow us to, even with the autosome, it'll be more like a Y and a mitochondrial thing, that we'll have things in the autosomes that are preserved over many, many generations. We just haven't tapped into that yet in genealogy.
Would you say that those...
To go along with that, Dave, I will also throw in specific autosomal SNPs that may have occurred 100, 200, 500, 1,000, 2,000 years or so ago. If we start mapping those SNPs like Rob does with the Y tree, you can do the same type of thing.
Yeah.
Yeah, we'll get into that when you come on my question about bringing ethnicity data as a Bayesian prior.
Yeah, or clan stuff. I mean, a lot of us, my mother's side is all Scotch-Irish, and I have all kinds of lines that stop in colonial times. And yet, like in my Irwin line, my second cousins have tested their big Y, and we know within a very small area in Scotland where we come from. And Rob, you showed it nicely with all those little gray bodies where you don't know who they are, but you know where they came from, and you know what those ancestors were, where they were, and what they were like.
So, David, would you say that the TADs that you're describing, when scientists are making a genetic map and determining how many centimorgans are on a segment, that's not taking into account the TADs, and so the centimorgan values we know are not actually indicative of how often a recombination might happen?
Correct. Chromosomes are different. There's different crossover rates on different chromosomes too. So there's more complexity here than the whole notion about randomness in Mendel. I almost made a slide, looking at Newton and Einstein and how different they were and how they looked at the world. And, Mendel looked at the genetics in one way, and what we have now is entirely different. And I'm a neurologist, and neurologists saw several inheritance patterns which were very peculiar. The one that everybody understands now is strictly maternal inheritance. We now knew that's, mitochondrial DNA mutations. But one of the other ones we observed was called parental effects. So if you have an inherited disease if you inherit Huntington's disease from your father, it's more severe than if you inherit it from your mother. And another disease, myotonic dystrophy, a muscle disease, if you inherit it from your mother, it's worse than if you inherit it from your father. So those were parental effects. We've known about those for decades. And it was not until we learned about epigenetics that we now understand that males and females are different. And the way they process DNA when they're making an ovum and sperm is different. And that explains those kind of parental effects. So what you're doing now, Brit, with trying to distinguish the path of inheritance is, I think, very important. And it's just a fundamental thing that's been largely overlooked in genetic genealogy.
I'd like to add a few more comments to this whole topic, too. I think my take, Brett, is that the centimorgans are still working, but there seem to be some very small subunits that are somehow better preserved. And the HLA region that Dave mentioned on chromosome 6 is certainly a classic one of those. I think if we looked at it too, we would see that there's a lot of homozygosity in certain little small regions of the genome, particularly for like Europeans or populations like that. And when you've got homozygosity for some of these shorter regions, those are going to be preserved even if there's a crossover, because there's no difference in the underlying DNA.
Yeah.
What kind of refinements can we expect to make in the future regarding our relationship predictors?
Well, I mentioned that one. I think we're going to have much more concrete examples of segments that are truly monophyletic. And so I think we're going to have the equivalent in the autosomes of what we now have on the Y and mitochondrial DNA. I think that's going to would be a revolutionary breakthrough.
I think I'm going to be a little more abstract and maybe pessimistic and think it's going to go more and more to the vendors, and they won't tell you exactly what they're doing. But like Ancestry has done and has shown they've done in publication, they will combine all the sources of data they have, which is considerable. Again, family trees and all the aDNA and all the details of that. And they're going to do a massive mashup and basically do full tree reconstruction, family tree reconstruction. Yeah, I don't think that's very far away.
Yeah, I was thinking about how, Rob, you said, using Y-DNA as an exclusion. So imagine a seg-CM prediction where the top probabilities by far are all paternal first cousin group, but Y-DNA doesn't match. Well, that becomes kind of a bad prediction unless the tool is taking into account Y-DNA, excludes all those, and recalculates the probabilities. Maybe now maternal-paternal is something that's very high for one of those relationships.
It comes down to data access.
Yeah.
Well, so that I'd like to speak to that, this locking up of data, and how does data going to be democratized? This has been a big problem in healthcare in particular, too, all the privacy, confidentiality, all this stuff. But it's really been mostly a problem with the vendors who have locked up their data, and the Epic won't talk to this one and they won't talk to the other ones. So it's been all these proprietary things that have locked up the data. When you think about what it's going to be like for our grandchildren and great-grandchildren, they should have access to all our DNA. I mean, there's no privacy, confidentiality, any of that kind of stuff. So I see in the future, this data is going to be democratized, and it will be used in a whole different way. I mean, if you think about the big data that's out there, I mean, we ought to be able to construct a genealogy in a matter of a few seconds from all of the data that's out there. If it was all democratized and connected together, it would be something that would be quite feasible.
Unfortunately, there are a lot of people, like most of the European Union, perhaps, or state of California, that will disagree with you on privacy basis. It's wherever they draw the line.
Well, but if you look at look at the GenBank, which is maintained at the National Library of Medicine. GenBank has I don't know, maybe several thousand, maybe 10,000 family tree DNA mitochondrial sequences. They've been submitted by people because they want to put their data out there to share with people. And I think that's going to become more and more common, that people will voluntarily put stuff out there. And the other thing is that the confidentiality of genetic information is going to sort of disappear. I used to talk to patients when I was still in practice who have a genetic disease. The point I would make to them is that we all have fatal genetic genes. It's just by chance that we happen to marry somebody who has the same one. So, I mean, I think the realization that we all have bad genes, everybody does, is going to make this confidentiality thing kind of disappear. And really what you want to do is you want to know what those bad genes are. You want to know what other people have done with that, and how can we get together. And that's part of what 23andMe is trying to do. I think that's going to become more and more prominent over time.
I'll agree with Dave in one sense. I remember there's a science paper, I'll extrapolate from it without explaining all my logic. A while ago, did the analysis of how forensic DNA works, okay? And this is maybe 5-6 years ago. They showed through a lot of what the data they could get that maybe 60% of European heritage Americans, you could find 60% of us in a forensic sense, meaning you have a cold case file, somebody does the DNA, 60% chance of finding somebody. It's probably up to 80% now.
It's more than that. It's more like 90%.
Okay.
Yeah.
Anyway, 80-90% can now be found. So in that sense, I agree that no matter what one may believe about it, in practice, this kind of forensic DNA sense, privacy is becoming a moot point.
Yeah.
What's
it's
amusing,
very hard to be a cook anymore.
Correct. What's amusing, in a sense of making jokes about the French, as anybody's English speaker will do, is that a lot of European DNA came over on the boat, a long time ago with us. So all that those giant American databases make, to some extent, increasingly a mockery of European forensic privacy rules, which is, so yeah, we're becoming one planet pretty quickly. I agree with that.
Yeah, about companies not sharing data, I think that's to be expected. They're
Absolutely.
Always going to go for the profit and hold on to their secrets. It's really up to us. We want to organize our data better and get large sets of data.
The profit motive, I agree, but remember, they're also constrained by privacy regs. They sell in Europe, they sell in California, I don't know where else in the world. And if they want to sell there, they have to abide by the rules. And that's fair enough.
Yeah, but they will have a huge advantage. Things like taking into account the Y-DNA, X-DNA, and all sorts of things. It'd be a lot better than somebody entering things into 8 different input boxes. It'd be so much better for a tool to be smart and take all those things into account.
Well,
not just that, they'll bring in hundreds of people. Yeah.
There are ways to anonymize data too, and that can overcome a lot of the privacy discussions you have. This has come up in healthcare a lot. I mean, there's a lot of standard data sets in healthcare that are anonymized, and they're very useful for doing analytics. We need that kind of thing in genealogy too. And to a certain extent, that's what you get with GenBank. And I mentioned those, maybe, I don't know how many there are, 1,000 family tree DNA things up there that you can't identify those people, yet they're available for studying mitochondrial sequences.
Yeah, that's probably true. But there was the 1,000 Genomes Project, which you may recall. They identified some of the participants in that using careful analysis of the genomic data.
Correct.
So I think that's kind of what Rob's. I mean, there's a full spectrum here of perspectives, and there's no easy, simple, right answer. I
Exactly.
think. And yeah, some of the data, particularly like mtDNA, you may not be able to figure out exactly who it came from, but when you're talking about a full genome sequence,
you can tell.
Or even just taking like what's going on with forensics or unknown parentage cases right now. The forensic genetic genealogists are being very successful with looking at SNP sets of 500, 600,000 or so and plugging them into Family Tree DNA, Family Finder, or into GEDmatch.
Yep, I stand corrected. I think that's true. There's another project people might want to look at, and it's called All of Us. It's a NIH-sponsored project that is trying to enroll 1 million people. It's kind of like the old Framingham heart studies that were, 10,000 doctors signed up and they were followed longitudinally. The All of Us, with those 1 million people, they're going to do full genome sequencing on you. They're going to get medical histories. You give them access to your medical records, and they're going to got scientists and every kind of discipline you can imagine involved with that. So it's something I think when I signed up, there were something like 400,000 already signed up. I don't know whether it's closed yet or not, but if you're at all interested, because they give you the data, they'll give you your full sequence data back and things. So it's All of Us, and you find it at a website and you could look at it.
Britain and Iceland have similar things. They have had for years. UK Biobank and Decode in Iceland are similar projects.
We've talked about this already a little bit too, but just how are we going to overcome endogamy and background relatedness when we do our relationship calculations?
So if you go to Graphs for Genealogy, you'll see a couple of blog posts on endogamy. Tim, you and I have started working on this with the Mennonites. Those blog posts are about French Canadians. It's very easy to get a whole different perspective on endogamy using a graph database. We can calculate all the relationships, all of the number of paths to a common ancestor. Most people, when they think of endogamy, they think about somebody's in different places on the family tree. They have different anatafel numbers. But in fact, the person's only in your tree one spot. The difference is in a graph, you can see it there. You just have 5 paths to the person instead of 5 positions in the pedigree chart. We can calculate the coefficient of relationship. The other important thing in endogamy is, and I've kind of coined the term, the most recent endogamous ancestor or union, most recent endogamous union. That's the place in your pedigree where the first place in your pedigree where a couple married who have a relationship, a known relationship. And their coefficient of relationship can be calculated. And then there's called a coefficient of inbreeding, which is that coefficient of relationship divided in 2 for every generation you have up until that point. And I think we're the only ones who are calculating coefficients of inbreeding using pedigrees in endogamous populations. And you can describe all of the paths very easily. We thought when we did this project that we would be able to find paths that could be uniquely identified. So could we find segments that just came down this path and not down that path? And I can tell you, we worked hard on that, and it is not possible. There's so much cross-linking that you cannot find a person who would isolate one path in those families. So that's a major issue. The thing that came up the other day that I was interested in is in those populations, we know that every endogamous population has certain diseases. The Mennonites have their set, the French Canadians have their set, Ashkenazis have their set. They're different diseases because different genes have come together in those populations. So the question, Britt, this is kind of a thing you might be able to help with, is how do you calculate, let's say we have this one common ancestor back there who had the mutation. Now we're looking at lots of paths down to this 7th generation down. How do we calculate sort of the odds that those people are going to actually have both copies of that mutant gene?
Yeah, I don't know. I haven't worked on a problem like that.
I think it's a problem that's solvable, and because actually that's another thing you could use. I mean, we haven't talked about that, and it's been kind of taboo. Like I've looked now at the GenBank 60,000 sequences of mitochondrial DNA, and I can find mutations for mitochondrial diseases in several thousand of them. So it's possible to do that now. Are those going to be markers that are useful, where we could start using some of those markers to document a path down a pedigree?
Some of you know the George Fry story of how they were able to pin a specific autosomal mutation that was autosomal dominant that was passed down from the early Puritan immigrant George Fry or his wife. I don't know that we've got it sorted out there, but all those people are at significantly higher risk for colon cancer. And so the LDS database was important for that. But that's the kind of thing you're talking about, Dave, is using some data set and knowing what the medical predictions are for some of these things. And I've been a strong advocate for like BRCA mutations and things like that we need to be sharing that kind of information as well, that people aren't left in the dark about their medical probabilities for certain things like breast cancer, ovarian cancer, colon cancer, etc. And I think rather than hiding behind a wall of privacy, we should be openly broadcasting that information to people who have that kind of information.
Yeah, Tim, you probably did the same thing or do the same thing I did when I was in practice. I mean, you diagnose a genetic disease and you talk to that family, tell your cousins and everybody else so that, we can head it off, or actually nowadays with prenatal testing and stuff, and I'm not talking about abortion, we can look at embryo, at ova and sperm and pick the ones that don't have the disease. So I agree with you. We should be making all of this, encouraging families to really propagate this information.
We're getting into medical DNA, which is fine or not. Tell me if I'm wrong. I thought I knew, maybe I don't, that the 600 or so thousand SNPs that we do when we buy a typical consumer autosomal test were selected as best possible to be neutral. Not code. No.
I disagree with that.
No, they're not at all. I mean, you can go to what's the
but it's a very different set of chips when you buy a disease predictor or a trait predictor.
Yes, but you can take your autosomal DNA from any of the vendors and put it up on this website, and it'll give you your risk.
Promethease.com.
Yes, exactly. Promethease.
You can and you'll get predictions out of any of the SNP sets you get from Ancestry is actually loaded with health-related ones. They particularly wanted to do it that way. 23andMe, of course, is loaded as well. Family Tree DNA and MyHeritage, they're trying to use kind of a more broader, the GSA SNP array, but that's still got health predictors in there too.
Yep. And it's not hard to match these things up. I've taken the SNPs that you can get and matched them up with what's on OMIN. OMIN is it's online inheritance in man. It's a database that you can actually download that has all of the known genetic mutations in man that have been cataloged, and you can map those to your DNA data. And I did this partly to see because I was concerned about pileup regions, because when you start talking about monophyletic segments coming down through many generations, well, we know about pileup regions in general, but there may be pileup regions within a family. So what I was interested in was, can you take the known pileup regions and see what's going on with the genes in that area? And it turns out that in the pileup regions that we know about, there's a lot of bad diseases there. And now I haven't done a thorough enough study to publish this or make this real, but the point I was trying to get at is there's stuff there that might make changes in those locuses unlikely, because people are going to die, the fetuses are going to die. And so there's a selection that happens from fatal mutations. And we know that happens in some situations very directly, that certain diseases are fatal in utero. And so there's a selection right there also against certain genes coming forward. But that also means it's favoring certain genes coming forward.
So, Dave, you brought up the coefficient of relationship a number of times in your initial presentation. So maybe just give your perspective on that. And you mentioned the coefficient of inbreeding as well. Maybe just a short synopsis on that from your perspective.
I think for what we're talking about today, it's a number. as opposed to, a notation like 1C, 1R. You can put a number to that. And is that going to be more beneficial than categorizing people by buckets? There's advantages to both of them, obviously. And even the first cousin once removed, that's not a stereotype person. They can have different paths to becoming first cousins once removed. I think my main message is about paths. And as I've worked with graph databases, that it's become very, very clear that in genealogy, it's the path that's important. And that's true in endogamy as well. I mean, you have all of these paths that connect you to a single ancestor. And if you have two people that are related, they have paths to many different common ancestors in their pedigree. I think we need to spend a lot more attention on paths, and a coefficient of relationship, coefficient of inbreeding are two ways to quantitate that, but they're not the only ones. And I'm particularly interested in maybe working with Brit on how do we get into the equation, the actual, paternal maternal into those analytics of the past.
We've talked about this a little bit in terms of knowing the sex of our ancestors. So this question, actually, Dave, I think was yours, was how does knowing the sex of our ancestors and the path to shared ancestors help us with our relationship predictions?
Yeah, so we definitely have seen more segments in relationships that have more maternal paths and much fewer segments in relationships that have more paternal paths. And we even see some predictions that show a mixture of the two, such as a paternal-maternal first cousin is the most likely relationship, which means it's more of the middle ground of the number of segments.
Hey, Britt, maybe you'd be the best person to talk about this in terms of the Watto tool, exactly how that tool works, and maybe your perspective on that tool in terms of how well it works.
Say you have an unknown match. If it's an ancestry, you'd have to ask your known family who have tested how much they share with that unknown match. So you build this tree. You have to put in your hypotheses for where you think that unknown match might fit into the tree. And it's going to give you probabilities. It actually does it not as probabilities out of 100%, but as odds. So high numbers are more likely and low numbers are less likely. But it's really just multiplying those probabilities together. It's also excluding things that aren't logistically possible. So 3 people match with close family type CM numbers, and all 3 match that way. You might be a half sibling of one and the other is your aunt or grandparent, but there are certain things that are not possible. For example, one is your aunt, the other one is not your nephew, because then they're sharing too much for that to be possible. They're no longer sharing in the close family range. So it's excluding those logical impossibilities. It's multiplying probabilities. It's not normalizing them back to 100%, and it's giving you that odds for the hypotheses you entered, or you can have a hypothesis suggested. But, it has those issues of multiplying probabilities. It would be much better if it was taking into account how much DNA coverage you had of an ancestor. And when you do DNA coverage, you actually want to use a multiple. For example, 2 siblings approximately have about 75% of their parents' DNA on average. So when you find out how many distinct segments are shared between a match with both you and your sibling, you actually want to multiply that value by 100 divided by 75 to approximate how much your parent would have shared with that match. If you don't, you're going to get a bad prediction. If there are 3 siblings, you multiply it by 100 over 87.5. If they're half siblings, you can actually find out how much of your parents' DNA is covered. You don't have to approximate. You can actually multiply it by the real ratio, because half siblings who share, say, 24% DNA, you know that you have 76% of your parents' DNA covered instead of 75%. Now you can multiply it by 100 over 76. A Watto tool that did that, coming up with the DNA coverage of the ancestor, using that instead of multiple siblings would be really good. And a Watto-like tool that used the number of segments would be extremely powerful. For close family predictions right now, SegCM likely does a far better job at telling you how a match is related to you than a Watto tool would, except for those logistical impossibilities that it's doing.
Yeah, I think you demonstrated that very nicely, Britt. That is an advantage. Wesley Johnston has done some mathematics. He's another mathematician who's done some stuff on DNA coverage. And we've actually implemented that in Graphs for Genealogists. So you can take all of your known matches and predict then, what kind of coverage you would have had of a given ancestor. And the way that becomes useful is if you say, I want to add another person to test, what impact is that going to have on your coverage of those more distant ancestors? And it's set up to do that too. You can add people and then recalculate. It takes less than a second to present the data. I mean, it does it all the way up. It shows you, here's all your matches, here's their parents and their parents and their parents, and it just meshes them all together as you go up the family tree and calculates the math at every step. It's quite robust. It's a way you can do those kind of calculations, which are difficult. If you do it manually, it'll take you a week to do it, but it does it in a couple seconds in a graph database.
Yeah, the principle is, I have 5 maternal siblings tested, so there are 6 of us. Testing another one, if there was one, wouldn't be as useful as if I had my first cousin test.
Exactly.
It can get a lot more complicated than that.
Yeah.
So it's good to have tools that do that for you.
Exactly. So next question has to do with distributions. I don't know if Dave, if you want to take this one on, but about whether we want to look at normal distribution, Poisson distribution, or power law, etc. Maybe Britt, you've got some background on this, but
if you look at the shared centimorgan project and the histograms that they have, and they look very normal distributed when you're looking at cousins, first cousins, and so on. But when you get to 4th and 5th cousins, they're skewed. So they have what are called a long tail on them. And those, the question is, what kind of distribution are they following, and what does that tell you about what's going on? Now, in graph theory, there's things called scale-free networks, which almost everything in biology emulates a scale-free network. Now, there's some debate about whether they're actually scale-free networks or some kind of related one. But the point is that in biology, almost no data is normally distributed. There are long tails in most data that you look at, and this would be like the $1 million baby. It would be a big example of that. They're rare, they're outliers, and the other concept is they're totally unpredictable. You cannot analyze to find those kinds of unpredictable things in a scale-free network kind of thing. It's one of the fundamental principles in graph theory. That's why I get concerned about some of the analytics that are based on normal distributions, as to they really apply when you get down the road, so especially when you get to more distant ancestors and things like that. Are we really having normally distributed data? And we know from what we know about genetics today that Mendel, all of what Mendel did and all of the subsequent derivative work was based on random normal distributions. And I just don't think biology behaves that way.
I've looked in some detail at the Y haplotree, also the MT haplotree, and certainly surnames. And if you want to call it a clade size, for lack of a better word, are very probably power law for a whole bunch of reasons. They are scale-free, depending how you sample them, which is a big hint. They're almost linear on log-log, although there's some, because you look at my research reports on my site, one of my most recent ones, there's like a ton of examples of scale-free, why that is, how the slope exponent depends on when you slice the tree. It's very, very clear for the Y haplotree because the data are so good.
Excellent. I'm glad to learn that, because that would be exactly what I would expect from a theoretical perspective, knowing what I know about graphs.
But but there's also extinction of lines and other things which are not normally covered in the graph theory.
Well, that's actually
it's built into all that.
That's actually the third pattern that neurologists observed was what's called anticipation. There are diseases that become more and more severe as you go down the pedigree. So the great grandpa he started when he was 80, and then 60 and 40 and 20, and eventually you get down to where it's before puberty and the disease dies out. And that anticipation, we didn't know what that was from either. We now do know. It's from trinucleotide repeat mutations, where the repeat length gets more and more each generation.
But,
So
but
it dies out.
just to be clear about extinction, I'm not talking about medical death. This is just the normal thing of we all know an aunt and uncle who have no kids or they only have daughters. I'm talking about that kind of why DNA extinction is extremely common. People don't generally know. This is more about genealogy, by the way, but in medicine, people don't normally realize how very common birth rates through most of European and not American colonial, that was exceptional history. Any given Y DNA lineage has about an 80% chance of dying out within 6 generations. That's part of what you have to bake in when you look at these distributions of why there's some Y clades that are huge, and there are a whole lot of them which are very, very tiny.
Yeah, but the question then, Rob, is why is that? I mean, it's not just sociology. There's I think it's biological basis for it.
I don't think it's selection in any disease sense. I think it's just random.
Well,
it's easy to simulate. Done a ton of that too. It's very easy to simulate on random Poisson models of birth death rates, and you get that. You get a power law. It doesn't take any science or medicine or anything. It's just random birth death rates. Give you the observed.
I understand what you're saying. I'm a biologist. I'm interested in what's the mechanism. How does that happen?
Yeah.
Okay, let's go on to our next question here, which has we've already kind of touched on this too, but in terms of the shared CM project, why is it skewed for more distant relationships? And for this one, I went to the shared CM project and pulled the data so we would have it for our reference today. Second cousins average 229 centimorgans. Third cousins average 73 centimorgans. Fourth cousins average 35 centimorgans, and fifth cousins average 25 centimorgans, which are way off from what we would expect, which would be for third cousins, we'd expect about 55 centimorgans, fourth cousins about 14 centimorgans, and fifth cousins about 2.5 centimorgans. So
is this just endogamy?
Well, I think that's part of it, but I think the other is that Blaine Bettinger only got data in many cases from people who had a known relationship. And so all the negatives were not submitted to him. All the people who were fifth cousins who did not share DNA with each other, those are out of his data set. So all he has is the positives rather than all the negatives.
Okay.
And that skewed his relationship predictors. And if people are using these and expecting to find 25 centimorgans shared on average with fifth cousins, they're going to be really kind of in sad shape.
Yeah, all those values for more distant cousins, they're all much higher than you would expect. And you can see what the theoretical value would be pretty easily. Cousins share 12.5% DNA on average. Divide that by 4, and you get the second cousin average, which is 3.125. You can divide that by 4 again to get the third cousin average. And for any percentage value, you can approximately get CMs by multiplying it by about 70 or so.
So do you think it's selection bias on Bettinger's part?
What's that?
Do you think it's selection bias on Bettinger's part, as Tim said?
Well, I do think it's hard to get people to submit data for a known cousin who shares 0 centimorgans of DNA. I think a lot of people are worried that they're not actually related in the way the tree says, or those people just those matches aren't on their radar because they're not seeing them.
This is another place for the coefficient of relationship, because in theory, it should be a linear relationship between that and your shared centimorgans, and it's not. And especially at those more distant relationships. So that's a very clear way of, with a single number to show what you're talking about, Brit.
Yeah, we either need to figure out a way to get those submissions, or to just substitute it with simulated DNA, or just based on the known amounts of how often we think cousins don't share, just adding those 0 values in.
Yeah, this is an interesting one to puzzle out, because of course it has to do with our perception of endogamy too. It might be much less than we think. If we're looking at Bettinger's data and thinking, wow, that was a lot of 18th century endogamy in the background.
Yeah, and that does affect, the more distant matches. The average will be higher than the theoretical average because of double relationships, triple relationships, endogamy.
Okay, this ties into the next question, which is, we all see a lot of distant matches. Brit, you talked about 5 centimorgan and 8 centimorgan distant matches. So what do you think's going on with some of these kinds of things?
With what we see when we get predictions from those?
Or just the fact that we have a lot of matches. For instance,
Exactly.
my dad has a match in Turkey. I think it was 12, 13 centimorgans. I think it could have been a crusader, maybe went there,
Yeah.
or maybe there's some other.
Put it this way: what would be the random background?
Yeah,
Yeah.
but
Yeah.
they're just long way back in time, and I think we just have to all accept that.
Yeah.
These 5 to 20 centimorgan matches in particular can be way past 10 generations.
Has anyone done a purely a simple pure random model to give us a theoretical background?
Well, we actually have some real data. I mean, like in the French Canadian one, we have, people who have common ancestors, and the advantage of them is that their common ancestors are many generations back. And when you look at the impact of that endogamy on their coefficient of relationship, it's minuscule. Siblings, instead of being 0.5, they might be 0.506. So there is a little impact on the endogamy, but it's really quite small for those distant relationships. And that's why I'm kind of interested in looking at specific genes like the diseases that come down, and as another way of looking at that.
Give you another interesting case, Dave. I only read this paper recently. It's a few years old now. An interesting estimate of the number of the effective population, which is not the same as the number of people, the effective breeding population that came over the Alaskan land bridge and begat all native North and South Americans was 70 people, 70. Talk about a bottleneck. So part of me is wondering, what's the background, if we will, in this context, what's the background of similarity of all Native Americans, North and South Americans, versus, let's say, Africans, who are the most diverse people on the planet? I don't know. It would be interesting to know.
So, Rob, to answer one of your questions, there was simulated data done by Speed and Balding. You might want to look that up sometime, that looked at centimorgans and the estimated relationship. And it was fascinating to me. I included in many of my presentations because there's a nice graph in there that shows like what your predicted relationship is going to be for any given centimorgan value. And Britt, you probably have looked
what's
at that.
What's the author again?
Speed
what's the name?
And Bald to it. I've got it in one of my presentations.
Yeah. And I think that was maybe 2013. So it's often quoted as saying that you can easily share DNA with a lot of matches who are 20 generations back. That, it's saying a lot of your, for example, 8 centimorgan matches would be 20 generations back. The simulations that I'm running are from PedSim. Scientists at Cornell developed that. And I think that paper came out in 2019. And I linked to that in my presentation. So I've gone back to 15th cousins and more distant. I can run 1 million pairs with PedSim at 15th cousin degree and not get a single match. I can do that 3 times and maybe get 1 match out of 3 million trials. So to me, the most recent research is showing that we don't generally share DNA with our 15th cousins unless they're related multiple ways over. Maybe, and they probably, like 15th cousins probably are related to you multiple ways over. So if you're related to one at 8 centimorgans, it's probably because you're related in so many ways.
If you do the coefficient of relationship, the odds that that's going to change that is really small. And that's why, again, I think there are these sticky things that do not follow those random distribution rules.
To me, that says that the DNA isn't inflated by the endogamy, hardly. It's negligible. But I think when you're related so many ways,
I'll throw another wrench in the works, a conceptual spanner in the works here, and I don't know exactly how to think about this. I'm sure everything you say is true. You're thinking about segments, the way we've been talking today. But I can also do look up a calculation of what percent Eastern hunter-gatherer am I, or ancient West Eurasian. In other words, these big, huge old groups. That's still the same DNA. It's my autosomal DNA, same 600,000 SNPs I got. So when you forget segments and go to the individual SNP level, to a different type of statistics, these patterns are very, very, very clear. And they persist, forget 15 generations, try going back 300. They're still there. So we haven't unified this realm yet of aDNA analysis. Someday we'll figure it out. or that paper by Hahn et al. I quoted in some of my questions, I think is figuring this stuff out at enormous scale.
Well, I think you bring up an important point. I mean, is that these vendors are creating the segments. I mean, they don't, you just don't come out with a segment. They're assembling the segments
between SNPs.
Yeah, exactly.
That's all. Yeah. It's inside. Yeah.
Yeah. Yes. So it's an artificial thing to a certain extent, which is one of the reasons I stuck with one vendor in my analytics, because I'm interested in the crossover points defined by the ends of the segment.
Right. They assume that this SNP and this SNP are the same. Ergo, the DNA in between is all the same.
Right.
Probably okay. But again, if you just look at the individual SNPs, you can do, like I said, Eastern hunter-gatherer or whatever it is. And that works fine back all the way to the dawn of humankind, or chimpanzees too, if you want to go back that far.
Yeah, this comes into our earlier discussions about whether we're using SNPs or whether we're using short segments, tags, as Dave was referring to there. So, all right, let's keep going. Let's go to another question here. Current methods use family tree relationships in DNA. What other specific types of data can be helpful? We've discussed this a little bit already. Predictions, geography, population groups, clan, tribe, religion,
throw surname in there. In some in patrilineal societies, it helps. Not perfect, but it helps.
Well, surnames is something I've done some work with too. In the graph that I create with those 250,000 matches, about half of them have a surname list in there. I can ask for a surname. I would put in different spellings and use a full text index, and I can find all of the matches in my family that I know who they are and people who share a surname with them. But even more importantly, I can search and say, in this line, I've got these 5 surnames that I know. So I put them all in there. And then I find matches who I have no idea who they are, who share like 3 or 4 of those surnames. And bingo, that just pops out. Those are really interesting people for me, because now I want to look at that match that prioritizes that match in my list. So I agree, surnames could be very helpful in this if you have a system.
Who is relatives named Sinnott here? Somebody. I just pulled up on one of my own tools. I have 6 on the Y haplotree where users can attach names. I have 6 spelling variations on Sinnott on the Y haplotree.
Right. Yeah, so I have that too. I mean, yes, you have to enter all the different spellings, and then you have to have an index that's full text, and we'll use wildcards and things like that.
Yeah, I got wildcards.
Exactly. Yep. But I agree. I think surnames is even in autosomal DNA studies, because that's what I'm talking about. I'm talking about people who match autosomally. If I got 250,000 people in there, how do I narrow that down? And one way to do it is with surnames. And somebody who matches and has 3 surnames in one line that's shared, then they become a high priority match.
But the general answer to your question, Tim, I think has to be that in some general sense, we are, or we should, or ancestry is, in some Bayesian sense, combining all this stuff. Geography.
I completely agree with that. And I think some commenter earlier had mentioned about the fact they knew the ethnicity of their ancestral line, and so they were able to split their paternal and maternal matches very easily. And many people can do that.
I can do that,
and I can do that, of course, because my dad's Mennonite, my mom's is early American. So that's the way that
it's easy. Is. Exactly. My parents have died a long time ago, but I'm perfectly phased because I know the German ancestor is all on one side. I can pull it all out.
All right, so let's go to the next question. Given the sizes of the various different ATDNA databases and sizes of family trees for modeling, fitting, et cetera, privacy regulations, are most of us, for practical purposes, limited to the analysis offered by the vendors? And otherwise, can we use private website tools? I think this is probably your question, Rob. So
let the others have at it.
Well, yeah, the answer is clearly yes. There's lots of things and it's not just website tools. Mine is not on the web. There are tools, third-party tools, and they can offer great value. I think what Rob and Brit have done are good examples of that. What I hope for was that somehow these would be interoperable in the future, so that as we get more and more into this stuff, that like I can generate a file, a CSV file that is perfectly usable at DNA Painter, but it has to be manually uploaded there. And so you have a choice as a developer. Are you going to develop your own DNA painter, or are you going to talk to Johnny Pearl and say, why don't you create an API so I can just send the stuff over and pop it up? So I think going to be the challenge is, of all of these third-party tools, is how interoperable are they going to be?
I'm more pessimistic, like I said. I think it's Brit said this as well. Between profit motive and privacy regulations, I think the vendors are going to choose to lock it up or be forced to lock it up to a significant extent.
Well, part of this depends. This is why when I talked in my early slide was on innovation and the need for standards. One of the things that was very key in healthcare was developing standards of interoperability, so that there are ways that you know exactly how to send this piece of data to somebody else in the system. Those are standard documents, there are standard ontologies and vocabularies, and it works very well.
I'm laughing because my memory goes back to a Roots Tech video of a couple of years ago, and there's a panel discussion about putting DNA information into the GEDCOM. And it's funny or sad.
But there actually is a LOINC. There is a LOINC system in medicine for formalizing genetic information.
Right.
Okay, there is a standard for how you do that.
But my comment is only it's rather comic and sad to watch some very bright people who are in charge of the GEDCOM standard basically completely fall over their own feet and punt the question and say, we can't do this.
They totally blew it within.
They did.
Right.
WikiTree tries to incorporate DNA, and it's difficult.
Well, what they're trying to do is they're trying to incorporate the mitochondrial and Y,
correct,
but not autosomal. So we've discussed this, Dave, you'll remember discussions we've had on this group about the need for large-scale chromosome maps where we designate and figure out as best as we possibly can where these small and large autosomal segments came from, and autosomal SNPs whenever we can as well. And I think that's going to be one of the big holy grails for the future, is figuring out where all these short and long segments came from.
Just a thought.
See, that's kind of a boil the ocean problem. I mean, we need to start with really basic stuff.
Well, and people are starting.
Just to change the culture of working with data standards.
Let me suggest one. I don't know that the standard exists, but the ancient DNA academic community is pretty good. Dave Reich's group at Harvard has published, not all their own, and collated a lot of others on an open access database for ancient DNA. Again, this doesn't have, for the most part, the privacy concerns, which is probably a big help. But there are, in some sense, evolving standards, implicit, I'm sure, in the sharing of basically BAM files, VCF files, whatever it is, in the ancient DNA community, which of course would work just as well, I presume, in the modern aDNA community.
I know that. There's also Ian has done a lot of work with Y chromosome. I mean, he's got, they've got databases so you can, and I've downloaded, 50,000 or I guess 5,000 or 6,000 Y VCF files and so on. So I'm talking about something different, which is, how do you start connecting the dots between these things? And you need data standards to be able to do that. I mean, healthcare, frankly, if you can look at simple ones, the finance industry did this decades ago, and they know exactly how to send data back and forth. And the insurance industry has standardized forms for every kind of claim you might want to make and transaction and so on. The legal profession is probably a decade behind the medical profession right now, but I've been working a little bit with guys at the Pritzker Law School at Northwestern because they're trying to emulate what we did in healthcare. And when you start talking about evidence, if you have standards for the storage and the tabulation of evidence, then we're going to open up a whole nother area in genealogy, because all of this extraneous data we're talking about now will suddenly become computable. And we'll be able to, put that into our equations.
Brit and Rob and Dave, I want to thank you all for being willing to participate. We've covered a lot of different topics and a lot of ground. And Brit, thanks in particular for your initial presentation on relationship calculators. And we'll look forward to seeing additional improvements for the SEG-CM tool and other incorporations into that, including maybe X and age or whatever else you want to bring to the table with that. So thanks again for everybody else's participation as well.